sorry, this is, and I'll have Smita give her background, but we are your team at Jumpcode Genomics. My name is Britt O'Keefe. I am a sales manager here at Jumpcode. We also have Jeremy Milford online, who is the University of Michigan rep that will help support any questions or concerns or comments people might have post event. And then Smita, who will be doing the presentation today, I will have her give you a quick intro and get started. So thanks for having us.
Hello, and thank you so much for having me. My name is Smita Pathak, and I'm here to talk to you today about improving signal-to-noise in single-cell sequencing for RNA-seq using CRISPR-clean depletion. The name of the product as it appears on our website is the CRISPR-clean single-cell RNA boost kit. Single-cell sequencing is useful for measuring the expression profiles of genes in heterogeneous cell populations. However, there are several key issues. As you can see here in the left panel, of the total amount of mRNA available in the cell, we are only able to capture 10 to 20% of it using traditional single-cell capture methods. This plot is from the Nature Communications paper by Grace Zhang et al., and it shows this poor capture efficiency. On the y-axis, we have the capture efficiency plotted by the ERCC GC content and transcript length. As you can see, the capture efficiency ranges between 0 and 20%. Now, directing your attention to the middle plot, we can see that single-cell data is sparse. This creates issues for computational processing downstream. We don't know if a 0 is a dropout, lost during the experimentation process, or an actual 0 in the expression data. This is typically caused by very small amounts of genomic material being amplified. And finally, on the right, we can see that single-cell data contains a high amount of noise. The counts per gene of 2 cells of the same type has a low R-squared. The large number of dropout events, as seen circled here, show that taking averages of means across cells is not best practice in interpretation. Expanding on that, as you can see in this plot taken from the paper cited at the bottom of the slide, the vast majority of single-cell data is noise. The red bars are the percentage of noise in each of the single-cell sequencing methods listed on the x-axis. In the purple dashed box, you can see the 10x genomics method highlighted, where almost 95% of the data is noise. The very small blue bars represent biological signal. We can see that as a percentage, the majority of the data coming out of single-cell sequencing is noise, not signal. Our method of removing signal that appears as noise from single-cell sequencing data is described here. Working with the 10x Genomics Chromium Next Gem single-cell 3' V3.1 kit, we remove unwanted reads using CRISPR digestion before PCR amplification in order to maximize the number of unique molecules that are recovered from single-cell sequencing. By removing unwanted reads, we are able to boost signal to noise, resulting in more coverage of areas of research interest. The total assay time for this approach, including the 1-hour incubation, is 2 hours, while hands-on time is only 45 minutes. The first step of our process is to make the RNP complex by combining the Cas9 protein with guide RNA. This takes about 15 minutes. The second step is an incubation with the RNP and the cDNA for an hour, followed by bead cleanup. This removes cut fragments from the sample. By now, you must be asking yourself, what are we removing? Great question. The content for depletion was designed by analyzing a cohort of publicly available single-cell 10x data from various sources. To expand on that further, let's take a look at this plot. In green, we have the total number of reads, or 100% of the reads that go on the sequencer. In blue, we have the reads that confidently align to the genome. And in purple, we have the percentage of genes that align to the transcriptome. Using Cell Ranger from the 10x platform, we are only taking the reads that align to the transcriptome for computational analysis. Thus, only 50 or 60% of the total reads from the sequencer are used for downstream processing. The regions targeted for removal are highlighted here. In light blue, we have non-polyadenylated ribosomal RNA, which are typically ignored and thus targeted for removal. In darker blue, we have the unaligned reads. These are consensus intervals that appear across all sample types that show up across data sets. In dark purple, we have 100 of the top highly expressed ribosomal and mitochondrial genes. These do not play a role in identifying important cell types and are typically removed from sequencing data informatically. We also targeted these for removal. And finally, in light purple, we have non-variable genes. These are highly expressed housekeeping genes where expression does not change across cell types. They play no role in distinguishing cell types across single-cell RNA-seq and are perfect targets for removal. This slide shows the in silico depletion as a percentage of aligned reads of 14 publicly available SRA datasets. It shows each of the buckets we have targeted for removal. 10% for ribosomal RNA that's non-polyadenylated, 33% for ribomitogenes in the transcriptome. Once we add the unaligned genes, we get to 51%, and finally, with the non-variable genes, we get up to a total of 58% of potential reduction in noise when combining all of the areas for removal. Now I'd like to show you some experimental data that we've collected in-house. This slide shows the setup of an experiment with PBMC samples. We wanted to compare the depletion conditions in triplicate against the control using the standard 10x workflow. Each of these libraries represent WTA splits of the exact same cell type. Here you can see the results of sequencing each of these libraries. On the left-hand side, we have the control compared to the depleted condition on the right. You can see that the distribution of reads has changed. In blue, we have the informative transcriptional reads, which has increased from 25% to 50% in the control to the depleted condition. We have done this by reducing the non-informative transcriptional reads, in green, from 38% to only 3% in the depleted sample. Another way of looking at the data is by looking at the number of UMIs per cell. In this plot, we can see that on a cell-by-cell basis, we are recovering more UMIs or molecules per cell. At a depth of 25,000 reads per cell, depletion recovers an additional 280 genes per cell, an additional 456 UMIs per cell, an additional 1,985 genes contributing to biological signal. Expanding on that, you can see here that the depletion rate increases the rate of identifying genes by 2.2 times. In the control population, we can see that for every 1,000 unique molecules recovered per cell, only 300 genes are identified. However, in the depleted condition, for that same 1,000 unique molecules, 650 genes are identified. In order to demonstrate that we did not perform any manual annotations of the dataset, we used a software tool that uses predefined legacy markers from published repositories containing greater than 1 million PBMCs. The software tool automatically identifies cell types in PBMC databases. The algorithm considers 3 key factors. First, genes that are up and down regulated. Second, the weight of regulation of each gene. And third, the frequency of abundance of each cell type within the entire population. Using this software tool, we were able to achieve unbiased clustering of cell types. Using the software tool described in the prior slide on both datasets, we were able to see 2 additional cell types in the depleted condition when compared to the control. Again, this required no manual manipulation from the data from our side. The additional cell types were recovered purely by removing the noise from the single-cell sequencing data. In these plots, you can see on the left, in purple dash, the control could not distinguish between monocytes CD14 and CD16. However, in the depleted sample, you can see that the CD16 monocyte is recovered as well as the dendritic cell. Expanding on this further, we used another software tool described in this Nature Protocols paper that takes user data and automatically determines cell types and communication pathways. We wanted to see if our depleted PBMC data would provide more information than the control condition. Here are the results comparing the depleted condition to the control condition using the software mentioned in the previous slide. The raw output is the total number of protein-ligand interactions that passed a statistical confidence threshold. On the y-axis, you can see the total number of interactions that passed the control. On the right, you can see rare cell types that are detected in the depleted condition, playing an important role communicating with immune cells in the blood. This critical slide shows that whatever biology you're interested in, there's more information to look at in the depleted condition when compared to the control. The same information is shown here in the network diagram. This is a visual tool to show how cells are communicating with each other. The lines in dark purple means a certain cell is communicating with its connection a lot. When looking at the control versus depleted condition, we can see that there are more and enhanced connections on the right. Now, considering a different question, we wanted to know, can we sequence less but recover the same information when comparing the control versus the depleted condition? Here we are comparing the control or undepleted condition at 27,000 reads per cell with the same control but downsampled the reads to half or 13.5 thousand reads and compared those to the depleted condition with only 13.5 thousand reads. What we can see is that we recover the same number of genes and UMI per cell in the depleted condition as we did in the control with double the number of reads. When you look at the control with only 13.5 thousand reads, we lose information. In the plots on the right, we can see that there's no bias in the distribution of data. There's almost a perfect overlay of genes per cell and counts per cell. Using the software to output UMAP plots, we are still able to recover 2 additional cell types even at half the number of reads compared to the control. This again shows the power of depletion. By removing the noise from the dataset, we're able to reduce sequencing by half and still see the benefit of higher signal, as evidenced by the recovery of these 2 cell types. So, in summary, CRISPR-Clean boosts informative reads in PBMCs by 100%. We see a 1.5x increase in genes per cell and UMIs per cell, and we are able to recover 2 additional PBMC cell types. Genes per cell and UMI per cell are comparable between full sequencing with standard prep and half sequencing with Jumpcode depletion. Now, I'd like to walk through a biological vignette that we were able to demonstrate through a collaboration with Dr. Amitabh Pandey, cardiovascular physician at Scripps in La Jolla. Dr. Pandey had collected smooth muscle cells from coronary and pulmonary arteries and was interested in studying why coronary arteries build plaque, while pulmonary arteries do not. He had already done 10x sequencing before and did not get a lot of signal out of the data. This slide describes the experimental setup of the collaboration with Jumpcode, where we compared control conditions to depleted conditions for both coronary and pulmonary cells. At a sequencing depth of 40 to 100 K reads per cell, depletion recovers more than 2 K additional UMI per cell. The reason we were able to do this is shown in the middle plot. the buckets of what we are targeting. We were able to redistribute 43% of the reads in the control samples to other areas of the transcriptome, boosting the signal to noise and recovering more UMI and genes per cell compared to the non-depleted samples. Here we again used a software tool published in this Nature Methods paper to calculate how cells are changing transcriptionally. We have one cell type, smooth muscle cells, taken from human coronary and pulmonary arteries. Because cells are changing constantly transcriptionally, we can see in the top two graphs that cells are switching between the two cell types. In the bottom two graphs, using the software tool, it automatically shows that for the control sample, all cells are changing in the same fashion and changing into its terminal fate. In the depleted condition, however, what the same software tool shows is that the cells are taking two routes to two different terminal states. One group is changing one way, and another group is changing in a differentiated way. This is not visible without depletion. This study shows the power of CRISPR-clean depletion in boosting signal-to-noise in a real biological study. Initially with the control sample, the study shows the cells are changing in the same fashion, but through depletion we can now separate and distinguish between how coronary cells are changing versus how pulmonary cells are changing. Researchers are interested in this because coronary arteries have the ability to generate plaque, but pulmonary does not. Being able to separate the two based on transcriptional markers is very important to understanding why there is a difference to begin with. So far, I have shown you data using the 10x 3' single-cell workflow, but you may be asking yourself, what other methods is this kit compatible with? This slide demonstrates that our kit is compatible with the PacBio single-cell iso-seq method. This method enables biologically relevant isoform detection at single-cell resolution. As you can see in the plot on the right, depletion drastically enhances novel isoform detection when compared to the control. The depleted conditions are in blue, while the control conditions are in orange. Our kit is also compatible with 5' RNA-seq, and the data on the right shows the enhanced number of genes recovered per cell. In blue, we can see the higher density of number of genes detected when compared to the control condition in orange. Finally, our kit is compatible with Visium spatial transcriptomics. This technology allows gene expression data to be recorded by location, as is shown on the figure on the left. Using our depletion technology in conjunction with this kit, you can see in the plot on the right that we are able to double the amount of informative transcriptomic reads. This is visible in the blue sections of the pie charts. To summarize, we have seen that depletion recovers more information per cell, increasing signal to noise. We have also seen that depletion statistically enhances biological interpretation. Additionally, depletion enables biological discovery that could not be found otherwise. And our kit can be applied to many different single-cell assays. Thank you very much for attending this webinar and for your attention.
And Smita, maybe I'll do a quick intro to the grant program and then we can take questions if that works.
Yeah, that sounds great.
Okay.
Sounds like a good plan. Thank you.
Okay, great. I'm going to share my screen now and hopefully it works. Okay. So we are going to announce this special grant program. So we
I'm not seeing.
You're not seeing it.
No. And you should be able to.
Try this one more time.
anybody else sees her screen and I'm not, please just speak up. Could be an issue on my end too.
Not seeing it.
You're not seeing it? Okay, I can just verbalize it and then we can send it out with a follow-up if that works.
Okay, yeah, we have confirmation that other people cannot see it as well. So thank you,
And
Chad.
that's fine. Yeah, thanks for confirming. So basically we have, and it's mostly just a written slide anyways, but we selected a few genomics core labs across the country to participate in this grant program. And we're excited to share it with your community of researchers here at the University of Michigan. It's primarily focused around any planned 10x runs, either Chromium or Visium. And if you are looking to gain more discovery power by using this tool, we definitely encourage you to apply. We also encourage folks who have potentially stored libraries or cDNA from previous 10x workflows to also apply. We will be supplying a link, hopefully in a follow-up email, that provides access to our grant submission landing page, where you can submit a 200-word paragraph describing the question you plan to address and how increased sensitivity will enhance your research. And the number of awardees will be determined by the core lab and based on a 24 sample kit. Since the core lab is graciously donating their resources to implement this kit, we do require submissions come from affiliates of the University of Michigan. And you will be responsible for your 10x library prep and sequencing components of the workflow. We encourage submissions to be submitted within the next 2 weeks. So ideally by July 8th, and we can work with the core lab for any exceptions around that time. So those are kind of the basic details of the grant program, and we hope to see some submissions, and we'll provide that link as mentioned in a follow-up email. So thank you so much for your participations, and we're ready for your questions.
If anybody has any questions, you can go ahead and unmute yourself and ask them. If you do not have questions at this time, that's fine as well. We will provide contact information for everybody so that you can email them as well directly. There is a question in the chat. Can you see it, Brett?
Yes, it says, what type of pipeline was used to run this? Smita, do you have? You're muted too.
Sorry, I was talking while muted. This was run with standard bioinformatic tools, so Seurat and Cell Ranger for top-level analysis and Seurat for more in-depth.
It has a question, SnakeMake, question mark?
I don't know SnakeMake was used particularly, but I can ask our bioinformatics team and get back to you.
Was there any ambient RNA filtering done?
So whatever filtering was done was exact same from the control to the Jumpcode depleted samples. And so each of those were treated identically.
And maybe I'll jump in with a question. Thanks so much for the presentation. Very interesting stuff. So a couple of questions. When you were identifying this sort of like non-variable RNAs for depletion, would you tell us just a little bit about how that was done?
So they looked at the top expressed non-variable genes. So the ones that are basically hogging up most of the sequencing reads, and they targeted those for depletion. By targeting the most expressed non-variable genes and knocking those down, they were able to redistribute those reads onto some of the lower expression RNA transcripts.
And that was sort of like non-variable across kind of like what?
Yes. So across.
But yeah, like what data were used to generate that?
Yeah, that's in that first slide I can pull up. So it was across the 14 sequence read archive 10x data sets.
Cool, thanks. And then this is also compatible presumably with 10x's add-ons like CellPlex?
That's right.
Sure.
Mhm.
Great.
And it's also compatible with other single cell sequencing workflows. We are just showing you data for the 10x3'.
Great. Thank you.
All right, and you guys said that they had, after we send out emails and stuff, you said everybody had about 2 weeks, beginning of July, to go ahead and submit for this.
Yep.
And then since I have, oh, there is a question that came in as well. Let somebody else read that.
If I use, yeah, no worries. If I use tissue sample for a single cell RNA-seq, which step should I apply CRISPR clean?
So it'll always come right before PCR. So this is after the cells are separated and you've done the first bit of the 10x workflow. So it doesn't really matter if you use tissue sample or whatever your sample type. We're always coming in with the depletion right before the PCR step. Does that make, okay, great.
Awesome. Yes, so I think 2 weeks from today looked about July 8th. And obviously we can work with the lab on, timing if there needs to be an extension or anything, but ideally 2 weeks is good. And then hopefully there's, projects that are in the pipeline that we could potentially work within as well. So.
Perfect.
Great.
Thank you. And I'll just remind everybody, so we're taking a little bit of a hiatus for July and August. We'll return in September, and we're hoping to have kind of a heavy hitter, ideally in September. So I'll be sending out emails with that, but I will also be sending out emails that have information on this grant program and Jumpcode. And so you guys will be getting lots of emails soon. So thank you so much for joining. Oh, somebody actually just came in with another. Have you published this technology anywhere?
So there are publications on the depletion technology, and the data that I presented with our collaboration with Dr. Pandey at Scripps is being published now. It should be, it's accepted. And so I don't know if it's out yet, but yes, the answer is yes. And if you would like links to that, we can absolutely share those. Okay, great.
There is also another.
Is there a Git repository we can look at? I would have to get back to you on that. I will check in with the team and I'll get back to you. That I don't know offhand.
And it looks like there's definitely interest in you guys sharing whatever publication information you can. So
Okay.
that would
Can
be
I ask,
appreciated.
what specifically you're looking for in the Git repository? Like, are you looking for the genes that we're targeting? And we have an Excel spreadsheet that I can share that has the genes that are targeted. That question was for Chad. Oh, okay. Genes and the pipeline that was used. Okay, perfect. Yep, I'll get that to you.
Great. All right, well, we can end it there. If there's any other follow-up questions, we're happy to help answer those. I don't know if we have any of our emails viewable, but we can include those in the follow-up email with the grant submission page. And we appreciate you all joining today. So thank you again. We look forward to working with you.
Thanks so much.
Looking forward to it. And yeah, if you just send me all the contact info for everybody I need, I will send it out to everybody.
Great. Sounds like a plan.
All right, perfect. Thank you everyone for joining. Have a wonderful summer.
Bye-bye.